Hello there, and welcome to the Sleepy Facts channel. I'm so glad you found your way here. Maybe you had a long day, or maybe you're the kind of person who likes falling asleep to glowing mysteries rather than counting sheep. Either way, tonight's journey is for you. Let your mind relax, your breathing slow, and your body grow light as particles drifting through dark water. Because tonight, we're exploring bioluminescent creatures, those living lights that turn the deep ocean into a galaxy of stars. We'll drift from the chemistry that creates their glow to the silent conversations they hold in flashes and pulses, discovering how darkness itself becomes a canvas for life's most beautiful expressions. Some of what you hear tonight may surprise you, or at least spark curiosity as you drift off. Before we begin, if this helps you unwind, consider liking and subscribing, one sleepy soul at a time. Now, let's begin. Bioluminescence is the production of light by living organisms through chemical reactions that occur within their bodies, a phenomenon found throughout the ocean but rare on land. This living light results from a molecule called luciferin, reacting with oxygen in the presence of an enzyme called luciferase, a reaction that releases energy as photons rather than heat, making it one of nature's most efficient light sources. The process wastes almost no energy as heat, converting nearly 100% of chemical energy directly into light, far more efficient than any light bulb humans have created. This cold light glows without warming the surrounding water, allowing creatures to illuminate without revealing themselves through temperature changes that predators might detect. The reaction happens in special cells or organs called photophores, tiny biological lanterns scattered across the bodies of countless marine species. Each flash or glow represents a carefully controlled chemical reaction, turned on and off through mechanisms as precise as any switch, the creature commanding light as easily as we command our fingers to move. The ocean, in its deepest reaches where sunlight never penetrates, becomes a theater of living light. Billions of creatures producing their own illumination in the permanent darkness. More than 90% of creatures living in the deep ocean possess the ability to create their own light, making bioluminescence not an exotic rarity, but the norm in the largest habitat on Earth. In the midnight zone, where darkness reigns absolute beyond a depth of 3,000 feet, bioluminescence becomes the primary means of communication, hunting, defense, and reproduction. The pressure of this environment, the cold, the endless night, all create conditions where producing light provides advantages that outweigh the energy costs. Species that cannot create light themselves often form partnerships with bacteria that can, hosting colonies of glowing microbes in specialized organs, creating symbiotic relationships where the bacteria receive shelter and nutrients, while the host gains the ability to produce light. This prevalence of bioluminescence in deep waters transforms our understanding of what constitutes normal ocean life, revealing that the sunlit surface zones we know represent only a tiny fraction of marine environments, while the dark depths, teeming with living lights, contain the majority of ocean volume and biodiversity. The deep sea glows with life, a constellation of creatures each carrying their own stars. The darkness not empty but full of light, generated from within. The color of bioluminescent light varies among species, but blue and green wavelengths dominate because these colors travel farthest through seawater, allowing communication and visibility across greater distances. Blue light, with its shorter wavelength, penetrates the ocean more effectively than red or yellow, making it the practical choice for creatures needing to be seen or to see in the aquatic medium. Some species produce green light, some blue-green. A few create yellow or even red bioluminescence, each color suited to particular functions and environments. The red bioluminescence produced by certain deep-sea fish creates a private spotlight. As most deep-sea creatures cannot see red wavelengths, their eyes adapted only for blues and greens. These red light producers can illuminate prey without alerting the prey to their presence, hunting with a flashlight that remains invisible to their victims, a biological adaptation as clever as any military night vision technology. The ocean filters light selectively, absorbing warm colors quickly while allowing cool blues to propagate. So evolution has shaped bioluminescent systems to produce wavelengths that work in water, creating an ocean filled with blue and green glows, stars that shine in colors determined by the physics of light transmission through the sea. The firefly squid, a small creature found in the waters around Japan, possesses thousands of tiny photophores covering its body, creating displays that rival any fireworks show when thousands of individuals gather in coastal waters during spawning season. Each squid glows with such intensity that fishermen once used the light from masked squid to illuminate their boats. The collective bioluminescence of spawning aggregations bright enough to read by, bright enough to cast shadows on deck. These squid produce light through symbiotic bacteria, housed in photophores distributed across their tentacles, mantle, and around their eyes. Each light organ independently controlled, allowing complex patterns and displays. The squid can modulate the intensity and pattern of their lights, creating waves of luminescence that ripple across their bodies, communicating with potential mates, or perhaps confusing predators with ever-changing displays. During the day, they descend to deeper waters, where their lights are less visible, rising toward the surface at night when darkness provides cover, and their bioluminescence becomes less conspicuous against the dim light filtering from above. They swim through the water like moving constellations, small vessels of light drifting with currents, their presence transforming ordinary seawater into something magical, temporary and fleeting, visible only to those lucky enough to witness their gatherings. Dinoflagellates, single-celled organisms drifting in the ocean's upper layers, create the phenomenon known as milky seas, or bioluminescent bays, where the water glows blue-green when disturbed by waves or swimming bodies. These microscopic creatures, each smaller than a grain of sand, produce light when physically jostled, a defensive reaction that may startle predators or attract larger predators that might eat whatever disturbed the dinoflagellates in the first place. The light they produce is brief, a flash lasting only a fraction of a second. But when billions of dinoflagellates respond simultaneously to the same disturbance, the cumulative effect creates glowing waves and sparkling wakes that trail behind anything moving through the water. In certain bays around the world, conditions become perfect for massive dinoflagellate blooms. The population density so high that any movement creates cascading light shows, turning the water into liquid stars. Swimmers moving through these bioluminescent bays trail galaxies of light, each stroke of their arms igniting blue-green sparkles, the water responding to their presence with luminescence that seems almost magical in its immediate response and brief duration. These tiny organisms, invisible individually, collectively transform entire bodies of water into living light shows, ephemeral and beautiful, nature's temporary art installations appearing and disappearing with the rhythms of plankton blooms and tidal movements. The anglerfish, one of the deep sea's most recognizable inhabitants, dangles a glowing lure extended from its head to attract prey in the darkness, where visual hunting would otherwise be impossible. This lure, called an esker, contains bioluminescent bacteria that the anglerfish cultivates in a specialized organ, providing the bacteria with nutrients and oxygen while they produce the light that draws curious prey close enough to capture. The relationship between anglerfish and their luminous bacteria is symbiotic and species-specific, with different anglerfish species hosting different bacterial strains, each producing slightly different light patterns or intensities. The anglerfish can control the light to some degree, adjusting its intensity or even darkening it completely by covering the esker with dark tissue, though it cannot turn off the bacteria's light production entirely. The lure waves gently in the current, or twitches through the anglerfish's own muscular control, imitating the movement of small prey, exploiting the curiosity or hunger of other creatures that investigate this mysterious light in the darkness. When prey approaches close enough, the anglerfish strikes with impressive speed, its enormous jaws and elastic stomach, allowing it to consume prey nearly as large as itself. The glowing lure dangles in darkness like a question mark, a mystery that draws the curious toward danger. Bioluminescence used not for communication or defense, but as a trap. Light becoming a tool for survival in an environment where food is scarce and every meal counts. The Hawaiian bobtail squid maintains a complex relationship with the bacterium Vibrio fischeri, which it houses in a specialized light organ on its underside, using the bacterial bioluminescence for a technique called counter-illumination. The squid hunts at night in shallow waters where moonlight and starlight filter down from above, casting shadows that would make the squid visible to predators looking upward from below. By matching the intensity of its bioluminescent light organ to the ambient light filtering from above, the squid eliminates its shadow, becoming effectively invisible when viewed from below against the faintly lit surface. This requires precise control, the squid continuously adjusting light output to match changing light conditions. A biological dimmer switch responding to clouds passing over the moon, to the phases of the moon, to the angle of starlight. The squid cultivates its bacterial partners carefully, providing them with nutrients and oxygen, while controlling their population through daily venting, expelling excess bacteria each morning, and allowing the population to regrow during the day. The relationship is established anew each generation, with juvenile squid born without bacteria and acquiring them from the environment, selecting Vibrio fischeri from among thousands of bacterial species present in seawater. This daily dance of light, matching this partnership between squid and bacteria, represents one of the most studied bioluminescent symbioses, revealing the complexity and precision that bioluminescence requires. Jellyfish produce some of the ocean's most spectacular bioluminescent displays, their translucent bodies acting as canvases for light that pulses and flows through them in waves and patterns. The crystal jelly, Aquaria victoria, produces green bioluminescence from specialized cells, lining the edges of its bell and tentacles, creating glowing rings and threads that make the jellyfish visible as luminous sculptures drifting through dark water. This jellyfish contributed more to science than beauty, as researchers studying its bioluminescence discovered green fluorescent protein, a molecule that revolutionized biological research by allowing scientists to tag and track specific proteins within cells, an application that earned a Nobel Prize decades after the molecule's discovery. The light production in jellyfish serves multiple purposes, possibly startling predators that grab them, or perhaps the flashing attracts larger predators that might eat whatever is attacking the jellyfish, creating a burglar alarm effect, where the victim's distress signal brings help indirectly. Jellyfish drift through the ocean like ghosts, their bodies mostly water, their substance minimal, yet capable of producing light that transforms them from nearly invisible to strikingly present, creatures of darkness that carry their own illumination, pulsing softly through the water in rhythms set by their simple nervous systems. The gulper eel, a bizarre deep-sea fish with an enormous mouth and a tail tipped with a glowing organ, uses its bioluminescent lure differently than the anglerfish, dangling the light behind itself rather than in front. This tail light may attract prey that approach from behind, allowing the eel to swing around and capture them. Or it may serve as a distraction, drawing attention away from the eel's actual head and vulnerable body. The gulper eel's mouth is disproportionately large compared to its body, capable of opening wide enough to swallow prey larger than the eel itself, its jaw articulating in ways that seem impossible, the entire front of the eel essentially transforming into a catching net. The stomach expands to accommodate large meals, essential in an environment where food is scarce and unpredictable, where going long periods between meals becomes routine. The eel itself is rarely seen by humans, living at depths where only specialized research vessels can reach, its entire existence unfolding in darkness and pressure that would crush conventional submarines. The glowing tip of its tail waves through the abyss like a signal buoy, the only visible part of the creature unless light is shined upon it. A small beacon in the vast darkness, purpose known only to the eel itself and perhaps to the prey it successfully attracts. Bioluminescent plankton serve as the ocean's base-level light producers, countless microscopic organisms creating a diffuse glow throughout the upper layers of the ocean at night. These planktonic creatures, including copepods, krill, and various larval forms, produce light primarily as a startle response to predation, flashing when disturbed in ways that might confuse predators or make them more visible to their own predators. The cumulative effect of billions of planktonic light producers creates a layer of potential bioluminescence throughout the upper ocean, invisible until disturbed, but ready to flash in response to any movement. Predators moving through planktonic clouds trigger cascades of light that may silhouette them, making stealth hunting more difficult, a defensive strategy that works not through the actions of individual plankton, but through the collective response of populations. These tiny lights, each lasting only moments, create an ever-changing constellation in the water column. Depth markers made of living light, layers of bioluminescence that shift with currents and migrations, rising toward the surface at night and descending during the day in one of the largest migrations on Earth. They drift with the currents, billions of individuals each capable of producing a brief flash, together creating a living light show that persists every night throughout the world's oceans, unwitnessed by human eyes except in the rare places where we venture into their realm. The lanternfish, small fish that may constitute the most abundant vertebrates on Earth by biomass, carry rows of photophores along their sides in species-specific patterns that serve as identification markers. Each species of lanternfish has a unique arrangement of these light organs, creating patterns as distinctive as fingerprints, allowing individuals to recognize potential mates of their own species in the darkness. Where visual identification through color or shape is impossible, these fish participate in the largest daily migration of animals on the planet, rising from deep waters at night to feed in the more productive surface layers, then descending again before dawn, a vertical journey of hundreds of feet that they repeat every single day of their lives. During these migrations, the collective bioluminescence of massed lanternfish creates layers of light detectable by sonar, false bottoms that confused early ocean explorers, who thought they had detected the seafloor at depths far shallower than the actual bottom. The fish travel in enormous schools, millions of individuals moving together, their combined lights creating enough illumination to be visible from submarines, the water around them glowing with their presence. They feed on plankton during their nighttime ascent, converting the ocean's smallest lifeforms into their own biomass, becoming in turn food for larger predators, their role in ocean food webs essential despite their small size. The patterns of lights along their sides flash and pulse as they swim, communication or identification continuing even in motion, each fish carrying its own code of lights through the darkness. Certain species of deep-sea shrimp produce bioluminescent clouds, ejecting glowing material from glands near their mouths when threatened, creating luminous smoke screens that confuse predators. While the shrimp escapes into darkness, this bioluminescent vomit, unpleasant as it sounds, represents a sophisticated defense mechanism, the glowing cloud mimicking the shrimp's size and shape for a few crucial seconds, while the actual shrimp jets away in a different direction. The predator, attracted to the bright cloud, investigates the decoy, while the shrimp escapes, the light serving as a distraction rather than illumination or communication. The chemistry of this defensive secretion must work quickly, producing intense light immediately upon release. The reaction triggered by mixing previously separated chemicals or by exposure to seawater. Some shrimp species can produce multiple clouds, recharging their bioluminescent glands between uses, while others appear to have a single-use defense, the glands requiring longer periods to refill after being emptied. These shrimp feed on detritus and plankton, occupying ecological niches where they process organic matter and become food for larger creatures, their bioluminescent defense allowing at least some individuals to escape predation and continue contributing to the deep-sea ecosystem. They hover in the water column or crawl along the seafloor, translucent and nearly invisible until threatened, at which point they become briefly brilliant, sacrificing chemical resources to produce light that might save their lives. The vampire squid, despite its ominous name, is actually a gentle drifter that feeds on marine snow, the constant fall of organic particles descending from the ocean's surface layers toward the abyss. This small cephalopod can produce bioluminescence from photophores scattered across its body, creating displays where light appears to drip off its arms like glowing tears or flash in waves across its skin. When threatened, the vampire squid performs an impressive defensive display, turning itself inside out to expose the spiny inner surfaces of its arms, while simultaneously triggering its bioluminescent organs, creating a spectacle designed to startle predators. The light show, combined with the sudden transformation in the squid's appearance, may confuse predators long enough for the squid to escape. Though, in the slow-motion world of the deep sea, escape happens gradually, rather than through quick sprints. The vampire squid's eyes are proportionally the largest of any animal, enormous orbs that gather the faintest traces of light in an environment where light is rare and precious. It drifts through the mid-water zone, passively collecting the organic debris that sustains it. Occasionally encountering other creatures in the darkness, its bioluminescence ready to deploy if needed, but otherwise conserved. The chemical resources required to produce light too valuable to waste on frivolous displays. The squid moves through darkness like a small ghost, its presence revealed only by the lights it creates. Living proof that bioluminescence serves many purposes beyond the simple production of light. The cookie-cutter shark, a small shark that takes circular bites from larger animals, uses bioluminescence in a particularly clever way, glowing along its entire underside, except for a dark collar that creates a silhouette resembling a small fish when viewed from below. This false target attracts larger predators who approach what they think is prey, only to have the cookie-cutter shark attach its suctorial lips to them and rotate its body, using specialized teeth to carve out a plug of flesh before dropping away. The shark's bioluminescence creates a disguise, rather than revealing its true nature. Counter-illumination that eliminates its shadow, but with a deliberate gap designed to attract rather than hide. The strategy works on a variety of large marine animals, including tuna, dolphins, and even whales, all of which bear circular scars from cookie-cutter shark bites, though the wounds are rarely life-threatening given the shark's small size. The shark itself is only about 20 inches long, yet it successfully parasitizes animals dozens of times larger through this deceptive use of bioluminescence. It swims in the ocean's twilight zone, where dim light from above creates conditions perfect for its shadow-casting trick, too deep for bright sunlight, but shallow enough that downwelling light creates the contrast needed for its false target to be visible. The shark has internalized its bioluminescent bacteria within photophores that line its belly, controlling the light output to precisely match ambient conditions, maintaining the shadow of its dark collar as the only visible feature when viewed from below. Bioluminescent bacteria exist freely in seawater and within the bodies of many marine animals, producing light through their own metabolic processes independent of host control in some cases. These bacteria glow whenever their population density reaches a threshold level, a phenomenon called quorum sensing, where the bacteria essentially count themselves and begin producing light only when enough neighbors are present to make the light display worthwhile. This collective decision-making means that individual bacteria floating in seawater don't waste energy producing light that would be too dim to matter. But once they colonize a host and reproduce to sufficient numbers, they begin glowing, providing their host with bioluminescence in exchange for nutrients and habitat. The bacteria live their own lives within their hosts, reproducing and dying in cycles that may not align with their host's needs, creating situations where host organisms must actively manage their bacterial populations to maintain desired light levels. Some fish and squid vent excess bacteria daily, reducing populations to manageable levels and ensuring fresh, actively growing bacteria that produce optimal light. The relationship between these bacteria and their hosts ranges from loose association to obligate symbiosis, where neither partner can survive without the other. the bacteria unable to persist long-term in the open ocean, and the hosts unable to produce light without bacterial assistance. This partnership between single-celled organisms and complex animals demonstrates how bioluminescence often requires cooperation, life creating light through relationships as much as through individual adaptation. The scale worm, a segmented worm found on the deep-sea floor, produces bioluminescent flashes from scales along its back when disturbed, creating a rippling light show that travels down its body like a wave. These scales, called elytra, are modified body segments that overlap like roof shingles, each capable of independent light production, allowing the worm to create patterns and sequences rather than simple uniform glowing. The purpose of this display remains uncertain, possibly startling predators, possibly serving as a burglar alarm that attracts larger predators to chase away whatever is threatening the worm, or possibly even breaking off the glowing scales as a distraction while the worm escapes. Some scale worms live in association with other deep-sea organisms, including tube worms and sea cucumbers, residing on their surfaces and possibly providing some defensive benefit through their bioluminescent capabilities. The worm crawls across the seafloor or over its host organism's surface, its segmented body flexing and bending, its scales catching any available light, but also ready to produce their own luminescence when needed. In the complete darkness of the deep sea floor, the worm's flashing serves purposes we can only guess at. The meaning of its light shows known only to the worm itself, and perhaps to the creatures that observe those displays. A language of light that remains largely untranslated by human researchers despite decades of observation. The midshipman fish, a shallow water species found along the Pacific coast, produces bioluminescence through rows of photophores along its sides that resemble the brass buttons on a naval officer's uniform, giving the fish its common name. These fish use their lights during courtship, males establishing territories in shallow water and producing humming sounds while simultaneously activating their bioluminescent organs, creating a multi-sensory display designed to attract females. The combination of sound and light makes male midshipmen remarkably conspicuous during breeding season, their courtship so intense and persistent that in some areas the humming from large aggregations of males can be heard by people living near the water, becoming a seasonal annoyance that prompts noise complaints. The bioluminescence in midshipmen is unusual for shallow water fish, as most bioluminescent species inhabit deep waters, where light production provides clear advantages. The photophores in midshipmen contain both the luciferin and luciferase necessary for light production within their own cells, rather than relying on symbiotic bacteria, giving them independent control over their displays. During the day, the fish hide in rock crevices or buried in sand, emerging at night to feed, and during breeding season to court potential mates, their lights flickering on as darkness falls, transforming the shallow seafloor into a field of glowing points, each male midshipman advertising his presence through light and sound, hoping to attract females swimming through the darkness above. Certain species of squid produce luminous ink, a bioluminescent version of the defensive ink clouds that many cephalopods use to confuse predators. When threatened, these squid eject a cloud that glows brightly in the darkness, creating a large, luminous mass that roughly resembles the squid in size and shape, a decoy that draws the predator's attention, while the squid escapes in a different direction. The chemistry of luminous ink requires mixing components that are stored separately within the squid's body. The reaction triggered by their combination as the ink is ejected, ensuring that the light production happens outside the squid's body rather than internally. This defense works particularly well in the deep sea, where any light is highly visible, and predators rely heavily on vision to locate prey. The sudden appearance of a large, glowing target proving too tempting to ignore, at least for the seconds required for the squid to jet away. The squid can produce multiple ink clouds if needed, though each ejection depletes chemical resources that take time to replenish, making the defense costly and not something to use frivolously. These squid belong to families that have refined ink-based defenses over millions of years. the addition of bioluminescence to their ink representing a further enhancement, an evolutionary innovation that improves the decoy's effectiveness in low-light and no-light environments. They swim through the midwater zone, hunting smaller creatures and fleeing from larger ones, their bioluminescent ink held in reserve for emergencies, a last resort that might mean the difference between survival and becoming a meal. The loose-jaw dragonfish possesses the ability to produce red bioluminescence, a rare trait in the deep sea where most creatures cannot see red wavelengths. This fish uses its red light like a sniper's laser sight, illuminating prey without alerting them to danger, as the prey's eyes cannot detect red light and thus remain unaware they are being observed. The dragonfish has special visual pigments in its eyes that allow it to see red light, despite most deep-sea creatures being blind to these wavelengths, giving it a private channel of illumination invisible to others, sharing its habitat. This is unusual enough, but the mechanism by which the fish produces red light is even more remarkable, as it appears to involve chlorophyll derivatives absorbed from the prey it eats, repurposing plant molecules for bioluminescence in a way that seems almost opportunistic. The dragonfish also produces blue-green bioluminescence from standard photophores, using different wavelengths for different purposes: blue-green for communication or possibly counter-illumination, red for hunting. Its jaw can unhinge and extend dramatically, allowing it to consume prey nearly as large as itself, essential in an environment where meals are infrequent and every feeding opportunity must be maximized. The fish drifts through the darkness like a predatory ghost, its red searchlight sweeping for prey, invisible to those it hunts until the moment of capture. Bioluminescence weaponized into a private hunting beam. The glowing sucker octopus, a small deep-sea octopus, possesses suckers lined with photophores that can produce light in complex patterns, though the exact purpose of these glowing suckers remains mysterious. The octopus may use these lights during hunting, the glowing suckers potentially attracting or hypnotizing prey, though without direct observation of hunting behavior we can only speculate. The lights might also play a role in communication with other octopuses, creating visual signals that carry meaning in the darkness, or they might serve defensive purposes, startling predators, or making the octopus appear larger or more dangerous than it actually is. Unlike many bioluminescent creatures that rely on bacteria for light production, this octopus appears to generate its own light through chemical reactions within specialized cells, giving it direct control over the timing and intensity of displays. The octopus moves through the water or crawls along the seafloor, its arms exploring crevices and surfaces, searching for food while its glowing suckers pulse and flicker, creating moving patterns of light that seem almost hypnotic to human observers, fortunate enough to witness them through the windows of deep-sea submersibles. The creature's intelligence, typical of octopuses, combined with its bioluminescent capabilities, suggests a sophisticated behavioral repertoire, though most of this octopus's life remains unobserved and undocumented, its behaviors known only to itself and to the dark waters where it makes its home. Bioluminescence requires significant energy investment, as producing the chemicals necessary for light generation draws resources away from other biological processes, like growth and reproduction. Creatures that maintain bioluminescent capabilities must constantly synthesize luciferin and luciferase, or in the case of those with bacterial partners, must feed and maintain populations of bacteria that themselves require nutrients and oxygen. This energetic cost means that bioluminescence must provide benefits that outweigh the resources invested in its production, a calculation that natural selection makes automatically by favoring individuals whose light production improves survival or reproductive success enough to justify the expense. In the resource-poor deep sea, where food is scarce and every calorie precious, the persistence of bioluminescence across so many species demonstrates its value. Proof that being able to produce light provides advantages that compensate for the metabolic burden it creates. Some creatures appear to regulate their bioluminescent capabilities based on conditions, reducing light production or allowing bacterial populations to decrease when food is scarce, prioritizing immediate survival over capabilities that might be useful, but are not immediately essential. The balance between the costs and benefits of bioluminescence varies with environment, life stage, and individual circumstances, creating selective pressures that shape when and how intensely creatures glow, the evolutionary economics of light production playing out in each individual's allocation of limited resources between competing demands. The comb jelly, despite resembling a jellyfish, belongs to a completely different group of animals and produces light through a different mechanism, its bioluminescence possibly having evolved independently from the systems used by other marine creatures. These gelatinous animals possess 8 rows of cilia that beat in coordinated waves, propelling the animal through water while creating iridescent light shows through the diffraction of light passing through the moving cilia, a physical optical effect distinct from bioluminescence. However, many comb jellies also produce true bioluminescence through chemical reactions, creating blue-green glows that pulse through their bodies in waves, light that appears to travel along their ciliary rows in coordinated displays. When threatened, some species flash their bioluminescence intensely, creating bursts of light that may startle predators, while others produce more continuous glows whose purpose remains uncertain, possibly attracting prey or serving functions we haven't yet identified. The comb jelly's body, composed primarily of water with minimal solid tissue, transmits light efficiently, the entire animal becoming luminous rather than showing discrete glowing spots, creating creatures that appear to be made of light itself when they activate their bioluminescence. They drift through the ocean like floating lanterns, their fragile bodies pulsing gently, their movements elegant and hypnotic, their bioluminescence adding to their otherworldly appearance, making them seem less like animals and more like manifestations of the ocean's own light-producing capabilities. The pineapple fish, a species found in Australian and Japanese waters, forms a symbiotic relationship with bioluminescent bacteria housed in light organs beneath its lower jaw, using the bacterial glow to illuminate the water in front of its mouth as it hunts at night. The fish can partially control the light by covering the organs with flaps of dark tissue, essentially creating shutters that allow it to turn its lights on or off as needed, though the bacteria continue producing light regardless of whether the fish chooses to display it. This relationship must be established each generation, juvenile pineapple fish acquiring their bacterial partners from the environment, selecting and cultivating the appropriate bacterial species from the countless other bacteria present in seawater. The process of establishing and maintaining this symbiosis requires specialized anatomy and behavior, the fish providing ideal conditions for bacterial growth while preventing other microbial species from colonizing the light organs and potentially outcompeting or killing the bioluminescent bacteria. During the day, pineapple fish shelter in caves and reef crevices, their bacterial lights visible but less conspicuous in daylight, emerging at night to feed on small crustaceans and other invertebrates, their chin lights illuminating potential prey hiding in reef structures. The glow from beneath their jaws gives them an unusual appearance, like fish carrying their own flashlights, practical illumination for hunters that work the night shift in reef environments, where darkness would otherwise make visual hunting impossible. The hatchetfish, a small silvery fish with an extremely thin body, uses bioluminescent photophores arranged along its belly to eliminate its shadow when viewed from below, a technique called counter-illumination that makes the fish nearly invisible against the faint light filtering from the surface. This fish possesses an impressive array of light organs, each individually controlled, allowing it to adjust the intensity and even the angle of light emission to match changing ambient conditions as it moves through different depths, or as surface light changes with the time of day and weather. The precision required for effective counter-illumination is remarkable, the fish continuously monitoring downwelling light with specialized photoreceptors on its back, using this information to adjust its ventral bioluminescence to create a perfect match that eliminates contrast between its body and the background light. During the day, hatchet fish typically descend to deeper waters, where the need for counter-illumination is less critical, rising toward the surface at night to feed. When darkness provides additional cover, and their light-matching abilities face fewer challenges, the fish's blade-thin body, seen from the side, presents minimal profile, while from below it becomes invisible through its bioluminescent camouflage, a two-pronged approach to avoiding predation that has allowed these small fish to thrive in the open ocean's midwater zone. They swim through twilight depths like living shadows, neither dark nor bright but perfectly matched to their surroundings, the ocean's own chameleons, creating invisibility through light rather than through color or pattern. Some species of ostracods, tiny crustaceans found throughout the ocean, produce spectacular bioluminescent displays during courtship, males releasing glowing clouds of luciferin into the water in species-specific patterns that females recognize and respond to. These underwater light shows can involve complex sequences, pulses timed with specific intervals, trails that spiral or zigzag through the water, each pattern unique to its species, and serving as a mating signal that prevents crossbreeding between similar ostracod species living in the same waters. The male ostracod releases its bioluminescent chemicals while swimming in particular patterns, creating luminous trails that persist for several seconds, visible to females who evaluate potential mates based on the display's quality and timing. This courtship through light requires the male to invest significant chemical resources in producing the luciferin he releases, making the display an honest signal of his condition and genetic quality. Females choosing males whose displays demonstrate the vigor and resources needed to produce impressive light shows. The ostracods themselves are smaller than rice grains, yet their bioluminescence can be visible from several meters away in dark water. Tiny creatures producing light intense enough to communicate across distances, many times their body length. They drift and swim through the water column, the males periodically creating their glowing signatures, brief artworks written in light that dissolve within seconds, courtship performed in ephemeral displays that exist only in the moment of their creation. The sea pen, a colonial organism related to corals, produces bioluminescence that travels along its length in waves when disturbed, creating rippling displays of light that flow from the point of disturbance outward along the colony's branches. This wave of light results from individual polyps, triggering their bioluminescence in sequence, each polyp responding to the signal from its neighbors, creating coordinated displays from organisms that have no central nervous system to orchestrate their responses. The mechanism appears to be chemical signaling between polyps. Disturbing one triggers a cascade that propagates through the colony, the bioluminescence following this chemical wave and making it visible as a moving front of light. The purpose of this display likely involves defense, the sudden appearance of light, possibly startling predators, or making the sea pen appear more formidable than it actually is, though the exact selective advantages remain subjects of research and debate. Sea pens anchor themselves in soft sediments on the seafloor, their feather-like structures extending into the water column to capture passing plankton and organic particles, their bioluminescence normally dormant unless something bumps or disturbs them. They transform portions of the seafloor into gardens of potential light, fields of organisms ready to glow when touched, creating unexpected illumination in places that are normally completely dark, living proof that even stationary, seemingly simple organisms can possess remarkable capabilities for light production and coordinated display. The Bermuda fireworm performs one of the most predictable and spectacular bioluminescent mating displays in the ocean, with females rising from the seafloor in spiral patterns while releasing bright green bioluminescent material, signaling males to approach and release sperm in clouds that also glow. This reproductive event happens with remarkable regularity, occurring at specific times after sunset on specific days following the full moon, the worms somehow synchronizing their reproductive readiness with lunar and circadian cycles. The female's glowing spiral creates a beacon in the darkness, visible to males who respond by swimming rapidly toward her while releasing their own bioluminescent material, creating expanding clouds of glowing sperm that fertilize the eggs the female releases at the apex of her spiral. The entire display lasts only minutes, but during those minutes, the water becomes filled with glowing spirals and expanding luminous clouds, multiple worms, often spawning simultaneously and creating submarine fireworks that have fascinated observers for centuries. After spawning, the worms return to the seafloor, where they spend most of their lives in relative obscurity, their dramatic bioluminescent displays reserved for these brief reproductive moments. The predictability of their spawning allows anyone with access to Bermuda's waters and knowledge of the timing to witness these displays, rare opportunities to observe bioluminescence in shallow, accessible waters rather than in the deep sea, where most light-producing organisms live their lives beyond human observation. The giant squid, perhaps the ocean's most famous mysterious creature, possesses the largest light-producing organs of any known bioluminescent animal. Photophores the size of lemons located behind its eyes. The function of these large light organs remains speculative, as giant squid are rarely observed alive, and their behavior in their natural deep-sea habitat remains mostly unknown. The organs might illuminate prey, allowing the squid to see in the darkness of the deep sea where it hunts sperm whales and large fish. Or they might serve communication purposes, allowing giant squid to recognize and signal each other in the depths. The size of the photophores suggests they produce significant amounts of light, possibly visible from considerable distances in the clear darkness of the deep ocean, though whether they produce continuous glows or brief flashes remains uncertain. The squid itself can reach lengths of over 40 feet, including its tentacles, making it one of the ocean's largest invertebrates, a creature whose very existence seems impossible, yet has been confirmed through occasional strandings, specimens captured in fishing nets, and rare encounters with deep-diving submersibles. The mystery surrounding giant squid extends to nearly every aspect of their biology, including their bioluminescence. These enormous cephalopods remaining enigmatic despite increased research efforts, their lives unfolding in darkness and depth that humans can barely access, even with advanced technology. They drift through the abyss like living submarines, their dinner plate-sized eyes gathering any available light, their own photophores ready to illuminate or communicate, their existence bridging the known and unknown, the documented and the mysterious. Bioluminescent fungi exist on land, producing greenish glows from their fruiting bodies or mycelia, creating the phenomenon known as foxfire that illuminates rotting logs and forest floors on dark nights. These glowing mushrooms and fungal networks use the same basic chemistry as marine bioluminescence, luciferin and luciferase, combining in reactions that produce cold light, though the specific molecules involved differ between terrestrial and marine systems. The purpose of fungal bioluminescence remains debated, with theories suggesting it attracts insects that might help disperse fungal spores, or that it provides no current advantage, but represents a vestigial trait maintained from ancestral species where it did provide benefits. Unlike marine bioluminescence, which appears in thousands of species, bioluminescent fungi are relatively rare, with only a few dozen species known to produce light, most of them found in tropical and subtropical forests, where conditions favor their growth. The glow from these fungi is usually quite dim, requiring dark adaptation to see clearly. But in forests at night, away from artificial lights, the glowing logs and mushrooms create an eerie, beautiful illumination that has inspired folklore and fairy tales throughout human history. The fungi fruit and glow according to their own rhythms, independent of lunar cycles or daily patterns, their light production continuous but variable depending on temperature, moisture, and the metabolic state of the fungus. They represent one of the few non-marine examples of bioluminescence, reminders that living light is not exclusively oceanic, Though the ocean remains its primary domain, the photophores of many deep-sea fish contain sophisticated optical structures, including lenses, reflectors, and filters that control and direct the light they produce, creating biological flashlights with focusing capabilities that rival engineered devices. These structures concentrate light into beams rather than allowing it to radiate in all directions, increasing the effective brightness and allowing the fish to illuminate specific targets or areas. Some photophores include color filters made of pigmented tissue that modify the wavelength of emitted light, allowing fish to produce different colors from the same basic bioluminescent reaction by filtering out unwanted wavelengths. The complexity of these organs suggests they evolved through many small steps, each slight improvement in light control, providing marginal advantages that accumulated over millions of years into the sophisticated systems present in modern species. The photophores must be maintained throughout the fish's life, the tissues constantly renewed, the optical elements kept clean and properly shaped, a biological maintenance challenge that requires resources but provides benefits that justify the investment. Looking at a deep-sea fish under bright light reveals rows and clusters of photophores that appear as dark spots or silver dots, betraying no hint of their light-producing capabilities until the fish activates them in darkness, transforming from drab and colorless to spectacularly illuminated, each photophore contributing to patterns and displays that carry meanings in the language of light that deep-sea creatures speak. Railroad worms, actually the larvae of certain beetle species, produce bioluminescence from organs arranged along their bodies, with different organs producing different colors of light simultaneously. These unusual creatures can glow green along their bodies while producing red light from organs on their heads, one of the few organisms capable of producing multiple colors of bioluminescence at the same time. The red light from their heads may serve as hunting illumination similar to the red lights of dragonfish, allowing the larvae to see prey without alerting them to danger, while the green lights along their body might serve warning functions, advertising their toxicity to potential predators. Railroad worms live in South American forests, crawling through leaf litter and hunting other arthropods, their bioluminescence serving them in terrestrial rather than aquatic environments, proving that bioluminescence can provide advantages in various habitats. The mechanisms by which they produce two different colors of light simultaneously involve different luciferins, or different modifications to the same basic chemistry, each system independently controlled, allowing the larvae to activate one, both, or neither, depending on circumstances. They represent one of terrestrial bioluminescence's most spectacular examples. Though like most bioluminescent organisms, they remain largely unknown to the general public, their glowing forms witnessed only by those who venture into their forested habitats at night. When their lights become visible, the larvae eventually pupate and transform into adult beetles that retain some bioluminescent capabilities, continuing to produce light throughout their life cycle. Though the adult displays are less dramatic than those of the larvae, certain species of parchment tube worms construct U-shaped tubes in sandy seafloors and produce bioluminescent mucus that they release when disturbed, creating glowing clouds that emanate from their tube openings, like luminous smoke signals. The worms themselves remain protected within their tubes, extending tentacles to feed on plankton and detritus, withdrawing rapidly when threatened, and simultaneously releasing their bioluminescent defense. The glowing mucus might startle predators, or it might serve as a burglar alarm, attracting larger predators that pursue whatever disturbed the worm. Indirect defense through recruitment of the predator's own enemies. The chemistry of the mucus must allow for quick light production, the reaction occurring immediately as the material is released, creating maximum impact while the threat is still present, rather than producing light seconds later, when the defensive value would be minimal. These worms live in dense colonies, their tubes covering areas of seafloor in tangles and overlapping structures, entire communities of worms, potentially releasing bioluminescent material simultaneously if the disturbance is widespread, creating zones of glowing water that must be conspicuous even in the dim light of shallow waters where some species live. The worms spend their entire adult lives in their tubes, building and maintaining these structures while filtering food from water flowing through them. Their bioluminescence held in reserve for emergencies, a defense mechanism ready to deploy at a moment's notice when danger threatens their sedentary existence. The salps, colonial tunicates that form long chains drifting through the ocean, can produce bioluminescence that travels along their interconnected bodies, creating light displays that span many feet as the signal propagates from individual to individual through their shared tissues. These gelatinous organisms form chains through budding, each individual remaining connected to its neighbors, creating superorganisms that can stretch enormous lengths through the water. When one member of the chain is disturbed, triggering its bioluminescence, the signal can travel through the colony, each individual lighting up in sequence, creating waves of light that flow along the entire chain's length. The purpose of this coordinated response remains uncertain, possibly serving defensive functions by making the chain appear larger or more formidable, or possibly helping coordinate the colony's feeding movements by providing signals visible to all members. Salps feed by pumping water through their bodies and filtering out plankton, efficient processes of the ocean's smallest organisms. Their bodies so transparent that their internal organs are visible through their tunics. They drift through the ocean's upper layers, neither fully individual nor fully colonial, their identity distributed across many connected bodies. Their bioluminescence, another shared capability emerging from their connected existence. The chains undulate gently with currents and swimming movements, flexible structures that can contract and extend. Their glowing displays, transforming them from invisible to spectacularly visible, showcasing another variation on bioluminescence's themes of defense, communication, and the unexpected beauty that emerges from biological light production. The stoplight loosejaw, another species of dragonfish, not only produces red bioluminescence, but also has organs that generate blue-green light, giving it a two-channel lighting system that it can use for different purposes simultaneously. The red light serves as its private hunting beam, invisible to most prey species but allowing the dragonfish to see them clearly, while the blue-green light might serve in counter-illumination or possibly in communication with other dragonfish that can see both wavelengths. The fish's jaw can unhinge dramatically, allowing it to attempt to swallow prey larger than its own head, an adaptation common among deep-sea fish, where meals are infrequent enough that passing up any potential food source becomes a luxury these fish cannot afford. The combination of red and blue-green bioluminescent organs, each with its own control mechanism and optical structures, represents a significant investment in light-producing capabilities, suggesting that the advantages provided by having multiple wavelengths available justify the energetic and developmental costs of maintaining these complex systems. The loose jaw drifts through the midwater zone, its bioluminescent organs dormant most of the time to conserve chemical resources, activated only when needed for hunting, or possibly for rare social interactions with other loose jaws. Its name derives from the jaw's extreme flexibility and the apparent lack of solid connection between the lower jaw and the skull. Though in reality, the jaw is attached by elastic ligaments that allow extraordinary extension while still returning to normal position after use, a mechanical solution to the problem of consuming large prey with a small mouth. Bioluminescent bacteria in the ocean can exist both freely in seawater and as symbionts within animal hosts, with some species apparently able to switch between these lifestyles, depending on opportunity and environmental conditions. Free-living marine bacteria produce light only when their population density reaches critical thresholds through quorum sensing, a chemical communication system that allows bacteria to detect the presence of many neighbors and coordinate their behavior accordingly. When free in the ocean, bacteria rarely reach the densities needed to trigger light production. But when they colonize a host's light organ and reproduce in that confined space, they quickly achieve populations dense enough to begin glowing, effectively turning on their lights as soon as they achieve suitable numbers. This ability to sense and respond to their own population density prevents wasted light production when bacteria are dispersed, while ensuring light production begins when bacteria find themselves in situations where glowing might benefit their host and thus themselves indirectly. The bacteria gain nutrients and protection from their hosts, while providing bioluminescence in return, a mutualistic relationship that benefits both partners and has evolved independently in many different lineages of both bacteria and host organisms. The bacteria's ability to produce light serves no obvious purpose when they are free-living, suggesting that bioluminescence evolved in the context of host relationships and persists in free-living forms as a vestigial capability, or because maintaining the genetic machinery for light production costs little when not actively being used. These microscopic organisms, invisible individually and only occasionally noticeable as free-living populations, become crucial components of ocean bioluminescence through their symbiotic relationships with animals, billions of bacterial cells working collectively to produce the light that makes their hosts visible or invisible depending on how that light is deployed. The umbrella squid, a small deep-sea cephalopod, maintains bioluminescent bacteria in a light organ on its mantle. But in addition to using counter-illumination like the Hawaiian bobtail squid, it also appears capable of ejecting bacteria-laden mucus as a defensive mechanism, creating glowing decoys similar to the luminous ink clouds of other squid species. This dual use of bacterial bioluminescence, both for camouflage and for defense, demonstrates how versatile these symbiotic relationships can be, the same bacteria serving multiple functions depending on how the host deploys them. The squid must carefully balance its bacterial population to ensure adequate light production for counter-illumination, while also maintaining sufficient reserves to produce defensive mucus if threatened, a management challenge that requires the squid to continuously cultivate and regulate its bacterial partners. The mucus-containing bacteria persists after ejection, continuing to glow as long as the bacteria survive and produce light, creating decoys that remain visible for minutes, potentially long enough to distract predators thoroughly while the squid escapes into darkness. The umbrella squid itself is small, only a few inches long, and feeds on tiny crustaceans and other zooplankton, occupying ecological niches in the ocean's midwater zone, where being small makes finding sufficient food challenging but reduces the risk of predation by limiting the range of predators large enough to bother hunting something so diminutive. They swim through the darkness with their bacterial partners, maintaining relationships that span the boundary between separate organisms, the squid and bacteria functioning as a unit. Despite their vastly different sizes and evolutionary histories, some species of brittle stars, relatives of starfish, produce bioluminescence from their long, flexible arms, using the light displays apparently as a defense mechanism to startle predators or break their focus during attacks. The arms can break off when grabbed, a defensive strategy common among brittle stars, and the severed arms continue to writhe and sometimes to glow for minutes after detachment, potentially drawing the predator's attention away from the brittle star's central disk, which contains most of its vital organs. This sacrificial defense, where the brittle star gives up an arm to save itself, works because the arms can regenerate over time, making them replaceable body parts worth sacrificing to avoid more serious injury. The bioluminescence travels along the arms in waves or pulses, creating moving patterns of light that might be hypnotic or confusing to predators, adding another layer to the defensive display beyond simple breakage and escape. Brittle stars typically hide in reef crevices and under rocks during the day, emerging at night to feed on detritus and small prey, their nocturnal habits, making bioluminescence more useful, as any light displays will be more visible in darkness. They move across the seafloor using their flexible arms in rowing motions, the central disc lifted slightly off the substrate, their 5 arms working in coordination to achieve surprisingly rapid locomotion for creatures that lack fins or jet propulsion. When threatened, They can sacrifice one or more arms while continuing to move with the remainder, escaping while their predator investigates the still active severed arms. A strategy enhanced by bioluminescence that ensures the decoy arms remain conspicuous and attention-grabbing even in darkness. The football fish, a type of anglerfish, uses its bioluminescent lure not just to attract prey but possibly also to attract mates in the vast darkness, where finding others of the same species presents a significant challenge. The female football fish, much larger than the male, dangles her bacterial lure in the water while drifting through the midwater zone, the light potentially serving dual purposes of drawing prey close enough to capture and advertising her presence to males searching for mates. Male anglerfish of many species are much smaller than females, and in some species become parasitic on females they encounter, biting into the female's body and fusing with her permanently, obtaining nutrients from her bloodstream while providing sperm for fertilization whenever she produces eggs. The female's lure thus becomes a beacon that serves multiple functions across different timescales, moment to moment attracting prey for feeding, and over longer periods potentially attracting males that ensure reproduction. The football fish's bulbous body, adapted for life in the deep sea, contains a large stomach capable of expanding to hold prey nearly as large as the fish itself, essential in environments where weeks or months might pass between meals, and passing up any potential food could mean starvation. They drift through the abyss like living fishing boats, their lures baited with light instead of meat, patient hunters waiting for prey to deliver itself to them, occasionally successful enough to survive and reproduce, passing on the genes for bioluminescence and for the elaborate lures that house and display it. The flashlight fish, found in shallow tropical waters, has the unusual ability among bioluminescent fish to turn its light on and off rapidly through mechanical means rather than chemical control. The fish possesses light organs beneath its eyes, filled with bioluminescent bacteria that glow continuously, but the fish can rotate these organs downward into pockets in its face, covering them with dark tissue and making the light disappear from an observer's perspective, then rotate them back to the exposed position to reveal the light again. This blinking behavior creates flashing patterns that the fish appears to use for communication with other flashlight fish, and possibly also to confuse predators by making the fish's position difficult to track as it alternately appears and disappears. The fish schools in small groups near reef structures, their collective flashing creating displays visible from considerable distances. Group communication through coordinated light signals. During the day, flashlight fish retreat into deep reef caves, their lights still glowing but hidden in the cave's darkness, emerging only at night to feed on zooplankton in the water column above the reef. Their bacterial partners require constant cultivation, the fish providing nutrients and oxygen to maintain healthy bacterial populations that produce the light the fish depends on for its survival and social life. They flash and swim through the reef's darkness like aquatic fireflies, their lights punctuating the night waters with signals whose exact meanings remain partially mysterious to human observers, but clearly carry information meaningful to the fish themselves. Certain deep-sea jellies produce bioluminescence in spectacular displays that involve hundreds or thousands of tiny light organs distributed across their bells and tentacles, creating what appears to be a living constellation when they activate their full light-producing capabilities. These displays might serve defensive purposes. The sudden illumination of so many points of light, potentially overwhelming a predator's visual system, or making the jelly appear larger and more formidable than it actually is. The jellies can control their bioluminescence, creating patterns that ripple across their bodies, or flashing all their photophores simultaneously for maximum impact, suggesting sophisticated neural control despite the jelly's relatively simple nervous system. They pulse through the water, their bells contracting rhythmically to propel them, their tentacles trailing behind, gathering any small organisms that contact them, their bioluminescence normally dim or inactive, but ready to blaze into brilliance if danger threatens. The transparency of their bodies means that light produced anywhere in their tissues can shine through surrounding tissue, making the entire jelly appear luminous rather than showing discrete glowing points, creating creatures that seem to be made of light when fully activated. They drift through the midwater zone between the surface and the abyss, occupying depths where dim blue twilight from above fades into complete darkness below their bioluminescence, allowing them to create their own light in this transition zone, where the sun's light loses its final battle with the ocean's absorbing depths. The ponelfish, a small, shallow water fish found in Indo-Pacific waters, maintains bioluminescent bacteria in a transparent muscle wrapped around its esophagus, creating a light organ that glows visibly through the fish's silvery scales. The fish can control the light's visibility by contracting muscles that either allow the light to shine through or cover it with opaque tissue. Though like other bacterial light organs, the fish cannot actually turn off the bacteria's light production, only hide or reveal it. Ponelfish use their lights during social interactions, possibly to maintain school cohesion at night or during courtship, when male fish display to attract females, their glowing bodies flickering as they swim through shallow coastal waters. The transparency of the muscles surrounding the light organ is remarkable, requiring special arrangements of muscle fibers and lack of pigmentation that would normally make muscle tissue opaque, an adaptation specifically for allowing light transmission that demonstrates the evolutionary modification of anatomy to serve bioluminescent display. These fish feed on small invertebrates and zooplankton, rooting through sand and picking organisms from the water column, typical inhabitants of coastal zones, where they form an important link in food webs, connecting microscopic prey to larger predators. They swim in schools, sometimes numbering thousands of individuals, their collective bioluminescence turning schools into moving clouds of light when viewed in darkness, though during daylight, their silver scales reflect sunlight, and their bioluminescence becomes invisible, relevant only after sunset. When the fish's daily routines continue under cover of darkness, the chain cat shark, a deep-sea shark species, produces bioluminescence from its skin, though unlike most bioluminescent fish that use their light for counter-illumination or communication, this shark's bioluminescence may serve to attract prey directly. The light is produced from numerous tiny photophores scattered across the shark's skin, creating a subtle glow rather than bright spots. The overall effect making the shark appear faintly luminous rather than displaying discrete light organs. The purpose of this whole-body bioluminescence in a predator remains somewhat mysterious, though it might make the shark resemble a mass of bioluminescent plankton, drawing prey fish that attempt to feed on what they perceive as an aggregation of small glowing organisms, only to find themselves within range of the shark's jaws. The shark itself is small by shark standards, about 2 feet in length, feeding on smaller fish and invertebrates, occupying a middle position in deep-sea food webs rather than being an apex predator. Its bioluminescence is relatively weak compared to some other glowing creatures, perhaps reflecting a balance between producing enough light to serve its purpose while not making the shark so conspicuous that it becomes vulnerable to its own predators. Chain cat sharks live on or near the seafloor at depths of several hundred to over 1,000 feet, regions where sunlight never reaches but where sufficient nutrients and prey exist to support complex communities of adapted creatures, each finding its own solution to the challenges of life in perpetual darkness. Some species of sea cucumbers, when threatened, can eject their internal organs as a defensive mechanism, and in bioluminescent species, these ejected organs glow brightly, creating a luminous sacrifice that distracts predators while the sea cucumber escapes. The glowing organs continue to writhe and produce light for several minutes after ejection, holding the predator's attention, while the sea cucumber, which can regenerate its lost organs over several weeks, withdraws to safety. This dramatic defensive display, combining both the shock of sudden organ ejection and the distraction of bioluminescence, appears to be highly effective, as bioluminescent sea cucumbers persist in ecosystems with numerous predators that would otherwise threaten them. The bioluminescence travels through the ejected organs in waves, creating moving patterns of light that might be particularly attention-grabbing, ensuring that predators focus on the decoy rather than the retreating sea cucumber. These echinoderms crawl across the deep sea floor, feeding on organic material in sediments, their mouths surrounded by tentacles that gather food particles and transport them to the mouth, living their lives in slow motion compared to more active creatures. They represent an important component of deep-sea ecosystems, processing organic matter and making nutrients available to other organisms, their contribution to ecosystem function far exceeding what their unremarkable appearance might suggest, and their bioluminescent defenses allowing them to persist despite being soft-bodied and seemingly vulnerable targets. The Atolla jellyfish, when attacked by a predator, produces a spectacular bioluminescent display called a burglar alarm, flashing rapidly in a pattern designed to attract even larger predators that might attack whatever is threatening the jellyfish. This defensive strategy relies on the principle that the ocean contains hierarchies of predation, and something attacking the jellyfish might itself be vulnerable to other, larger predators. The jellyfish's flashing creates a beacon that can be seen from considerable distances in the deep sea's darkness, essentially calling for help by advertising the presence of potential prey to any predator large enough to threaten the jellyfish's attacker. The display involves rapid pulses of blue light traveling around the jellyfish's bell in waves, creating a pinwheel effect that is highly conspicuous, the marine equivalent of an emergency siren. Whether this strategy successfully saves individual jellyfish from predation is difficult to study, but the persistence of the behavior across Atolla jellyfish populations suggests it provides survival advantages that outweigh the energy costs of maintaining bioluminescent capabilities. The jellyfish drifts through midwater zones, trailing tentacles that capture small prey, living a relatively passive existence, punctuated by occasional encounters with predators. Their burglar alarm defense system, held in reserve for these dangerous moments, when survival depends on attracting attention rather than avoiding it. The mechanisms by which organisms control their bioluminescence vary widely, from simple chemical mixing that occurs automatically when tissues are damaged, to sophisticated neural control that allows precise timing and patterning of light displays. Some creatures produce light reflexively in response to physical disturbance, the mechanical stress of being touched or jostled, triggering reactions that release light as an automatic consequence, requiring no decision or neural processing. Others possess complex nervous systems that can activate specific photophores individually or in patterns, creating displays as controlled as any deliberate movement, the organism choreographing its lights with the same precision it controls its fins or tentacles. The difference between reflexive and controlled bioluminescence reflects different evolutionary solutions to different ecological challenges. Simple defenses requiring only automatic responses, while communication and hunting benefit from precise control over timing and intensity. The neural pathways that control bioluminescence in species with sophisticated light organs remain subjects of ongoing research. Scientists working to understand how signals travel from the brain to photophores, and how organisms integrate visual feedback about ambient light conditions with their control of light production. These control systems, whether simple or complex, represent biological solutions to the challenge of turning light production on and off, managing a chemical reaction that might otherwise occur continuously or not at all, giving organisms agency over when and how they glow. The efficiency of bioluminescence far exceeds any artificial light source humans have created, with nearly all the energy from the chemical reaction being converted directly into light, rather than wasted as heat. This cold light production allows even small organisms to produce visible illumination without raising their body temperature or creating thermal signatures that might alert predators to their presence. The luciferin-luciferase reaction that produces most bioluminescence approaches 100% efficiency, meaning that essentially every molecule of luciferin that reacts produces 1 photon of light, an ideal conversion that human technology can only dream of achieving. This efficiency is essential in the deep sea, where energy is scarce and organisms cannot afford to waste calories on inefficient processes. Every bit of food consumed being allocated carefully among competing physiological needs. The lack of heat production also means bioluminescent organisms can produce light without disrupting the thermal structure of the water around them, maintaining their camouflage against predators that might detect temperature differences even in the dark. Scientists studying bioluminescence hope to harness these efficient systems for human applications, developing artificial lighting that mimics the efficiency of biological systems, though recreating the precise molecular conditions that allow such efficient energy conversion proves challenging when working with artificial materials rather than living tissues, evolved specifically for this purpose. Some deep-sea fish possess light organs on stalks that extend from their bodies, positioning the bioluminescence at a distance from their actual body, potentially confusing predators about the fish's location and orientation. These stalked photophores might make predators strike at the light rather than the fish's body, similar to how anglerfish lures attract prey, though in this case serving a defensive rather than offensive function. The stalks themselves are flexible extensions of tissue, controlled by muscles that allow the fish to move them somewhat independently of body movements, waving the lights in patterns that might enhance their effectiveness as decoys or signals. The evolution of these structures required significant anatomical modification, blood vessels and nerves extending into the stalks to support and control the photophores at their tips, developmental investments that suggest strong selective advantages for having externally positioned light sources. The fish swim through darkness with their lights extended like antennae or feelers, the bioluminescence potentially helping them detect obstacles or prey through some mechanism we don't fully understand, or possibly serving purely communicative or defensive roles. These stalked lights create appearances stranger than any science fiction creation. Fish whose anatomy seems designed by artists rather than evolution, yet perfectly functional for their deep-sea existence. Each unusual feature representing a solution to specific challenges that would be invisible to us without understanding the context of deep-sea ecology. The siphonophores, long colonial organisms that can extend over 100 feet through the water, produce bioluminescence from various specialized polyps along their length, creating light displays that travel through the colony in coordinated waves. These creatures, related to jellyfish, consist of many individual polyps, each specialized for different functions, such as feeding, reproduction, or bioluminescence, all connected and sharing resources throughout the colony. The bioluminescent polyps can trigger sequentially, creating moving patterns of light that flow along the siphonophore's immense length. Displays whose purpose might involve defense, communication with other siphonophores, or possibly coordinating the colony's own activities through visual signals. The scale of these organisms challenges our concepts of what constitutes an individual, as each siphonophore is simultaneously one organism and many, a colony that functions as a unit despite being composed of separate, though connected polyps. They drift through the ocean's midwater zones, their bodies forming curtains or strings that trail through the darkness, tentacles extending from feeding polyps to capture prey, while bioluminescent polyps stand ready to activate if the colony is threatened. The coordination of bioluminescence across such vast distances within a single organism or colony suggests sophisticated communication systems within the siphonophore, chemical or electrical signals, propagating through shared tissues to orchestrate responses that span many feet, creating unity from multiplicity. The squid Wattersonia scintillans, commonly called the firefly squid, possesses one of the most elaborate bioluminescent systems known, with 3 different types of photophores serving different functions, some involved in counter-illumination, others in communication, and still others whose purpose remains uncertain. The largest photophores, located near the squid's eyes, produce intense flashes visible from considerable distances, possibly used for communication with other squid, or perhaps to illuminate prey during hunting. Smaller photophores covering the squid's arms may help with prey capture, by attracting small creatures toward the tentacles, while the ventral photophores clearly serve counter-illumination, eliminating the squid's shadow when viewed from below. This multi-layered bioluminescent system, with different organs serving different purposes, represents significant anatomical and physiological investment, the squid dedicating substantial resources to maintaining 3 separate types of light-producing structures. The squid itself is small, only a few inches long, yet despite its size, it produces some of the most complex bioluminescent displays in the ocean, proof that elaborate capabilities need not require large body size. They gather in enormous numbers during spawning season in Toyama Bay, Japan, creating underwater light shows as thousands of squid flash and glow simultaneously, their collective bioluminescence so intense it can be seen from shore, attracting tourists and researchers who come to witness this annual spectacle of living light. Bioluminescent organisms face the challenge of protecting their light-producing organs from their own immune systems, as the bacteria or chemical compounds involved in bioluminescence might otherwise be recognized as foreign and attacked. In species with bacterial symbionts, the host must maintain tolerance for these foreign organisms, suppressing immune responses that would normally eliminate bacteria from internal tissues, while simultaneously defending against pathogenic bacteria that might attempt to colonize the same light organs. This selective tolerance requires sophisticated immune regulation. The host's defenses, distinguishing between beneficial light-producing bacteria and harmful species, maintaining one population while excluding others. The mechanisms by which hosts achieve this selective tolerance remain subjects of research, involving complex chemical signaling between host tissues and bacterial partners, mutual recognition systems that have evolved over millions of years of symbiotic relationships. In species that produce their own light through cellular chemistry rather than bacterial partners, the challenge involves managing highly reactive chemical compounds that could damage tissues if not properly contained, requiring specialized cellular structures that isolate light-producing reactions from other cellular components. These challenges highlight that bioluminescence, for all its advantages, comes with costs and complications. Organisms needing to develop supporting systems that allow light production without causing self-harm. Evolution, solving problems that arise from innovation as surely as it creates the innovations themselves. The distribution of bioluminescence throughout the ocean follows patterns related to depth and habitat, with bioluminescence becoming more common as depth increases and light from the surface fades into darkness. In the upper sunlit zones, bioluminescence is relatively rare, found mainly in organisms that are active at night when their lights won't be overshadowed by sunlight. In the twilight zone, between 600 and 3,000 feet deep, bioluminescence becomes increasingly common, with the majority of species possessing some form of light production. In the midnight zone below 3,000 feet, where darkness is absolute, bioluminescence reaches peak prevalence, with nearly every species capable of producing or hosting light-producing bacteria. This depth gradient reflects the increasing importance of bioluminescence as natural light disappears, light production becoming not an exotic trait, but a standard feature of life in perpetual darkness. The pattern breaks down near the seafloor in the abyssal zones, where many bottom-dwelling creatures lack bioluminescence, despite living in complete darkness, perhaps because bioluminescence offers fewer advantages to organisms that live on solid surfaces where tactile and chemical senses become more important than vision. Understanding these distributional patterns helps explain why bioluminescence evolved and where it provides the greatest advantages, revealing the ecological logic behind one of nature's most beautiful adaptations. Some species of arrow worms, small predatory zooplankton, produce bioluminescence in their guts. After consuming bioluminescent prey, the consumed organism's light continuing to glow for a time within the arrow worm's transparent body. This creates the unusual situation where predators become temporarily bioluminescent, not through their own light production but by eating glowing prey, inadvertently advertising their position to their own predators through the lights of their meals. This might represent a form of indirect defense by the prey, their bioluminescence continuing to serve protective functions even after death, by making their predators more visible and vulnerable. The arrow worms themselves are fierce hunters. Despite their small size, capturing prey with hooks that surround their mouths, feeding on copepods and other small creatures that drift through the plankton, their transparent bodies allowing them to be nearly invisible hunters when they're not glowing with consumed prey. The phenomenon of prey bioluminescence persisting after consumption highlights the complex ways that light production influences predator-prey interactions, the effects of bioluminescence extending beyond the organisms that produce it to affect those that consume them, creating cascading influences through food webs as light gets transferred through consumption and predation. The discovery of bioluminescence in terrestrial environments beyond fungi and fireflies reveals that light production has evolved repeatedly across many different groups of organisms, including certain snails, earthworms, and millipedes that produce glows under specific conditions. These terrestrial examples of bioluminescence are generally dimmer and less well studied than marine examples, partly because they're less common and partly because terrestrial bioluminescence often occurs in remote or difficult to access locations. The railroad worms and glowworms of various regions represent the most spectacular terrestrial displays. Their lights bright enough to be easily visible and sustained enough to serve obvious functions in attracting prey or mates. The chemistry of terrestrial bioluminescence sometimes differs from marine systems, using different luciferins or different enzymatic pathways, suggesting independent evolution of light production in terrestrial lineages rather than inheritance from a common luminous ancestor. The fact that bioluminescence has evolved multiple times independently in both marine and terrestrial environments suggests that producing light provides significant advantages wherever organisms find themselves in darkness. Evolution, repeatedly discovering chemical pathways that convert metabolic energy into visible illumination. These multiple independent origins of bioluminescence demonstrate convergent evolution, different lineages arriving at similar solutions to similar problems, the physics of light and the chemistry of life, creating constraints that guide evolution toward comparable outcomes, despite starting from different ancestral conditions. The relationship between bioluminescence and vision creates a feedback loop, where the evolution of light production drives the evolution of better eyes to detect that light, which in turn drives the evolution of more sophisticated bioluminescent displays. In the deep sea, where bioluminescence is the primary light source, eyes have evolved to become extremely sensitive to the blue-green wavelengths that most bioluminescence produces, some deep-sea fish having eyes capable of detecting single photons, the smallest possible units of light. This extreme sensitivity comes with trade-offs, including increased vulnerability to damage from bright light and reduced ability to see fine details. The eye is optimized for detecting faint glows rather than resolving sharp images. The arms race between light production and light detection shapes both systems. Organisms that can see bioluminescence better, having advantages in detecting prey, predators, or mates, while organisms that can control their light more precisely can better manipulate what others see. This coevolution between signaling and detection creates complexity in both systems, each advancement on one side, selecting for improvements on the other, driving the elaboration of both bioluminescent organs and visual systems toward greater sophistication. The deep-sea darkness, far from being empty of visual information, contains rich visual ecology based on bioluminescence. Creatures seeing and being seen through light they themselves produce, creating a visual world independent of the sunday. Certain species of octopuses found in deep waters produce bioluminescence from suckers on their arms, though whether they use these lights for hunting, defense, or communication remains unclear. The octopuses can control the lights individually, creating patterns that might mesmerize prey or confuse predators, or possibly communicate information to other octopuses in a visual language we haven't learned to read. The The suckers themselves serve their normal functions of gripping and tasting, the bioluminescence adding another dimension to their capabilities without interfering with their primary purposes. The octopus's well-known intelligence, combined with control over bioluminescent displays, suggests these animals might use their lights in creative and flexible ways, adapting their patterns to different situations rather than relying on fixed behavioral programs. Observing these octopuses in their natural deep-sea habitat proves extremely difficult, limiting our understanding of how they actually use their bioluminescence in the wild. Most of what we know coming from laboratory studies or brief glimpses from submersibles that can only observe for short periods before battery limitations force them to surface. The mystery of the glowing octopus arms remains largely unsolved. These intelligent creatures, keeping their secrets in the darkness where they evolved, their bioluminescent capabilities, hinting at behaviors and interactions we can only speculate about until better observation methods allow sustained watching of their natural behavior. The cost of producing luciferin varies among organisms, with some manufacturing their own through complex biochemical pathways, while others must obtain it through diet, consuming prey that contain luciferin or its precursors, and sequestering these compounds for their own light production. Organisms that manufacture their own luciferin face the metabolic costs of synthesizing these complex molecules, dedicating enzymes and energy to production pathways that serve only bioluminescence. Those that obtain luciferin through diet face different challenges, needing to consume sufficient quantities of the right prey species, and having to stop glowing if their diet changes or if prey becomes scarce. This distinction between dietary and manufactured luciferin creates different ecological dependencies. Diet-dependent organisms being more vulnerable to changes in food web structure that might eliminate their source of luciferin, while luciferin manufacturers pay ongoing metabolic costs, regardless of whether they're actively using their bioluminescence. Some species appear to obtain luciferin from their bacterial symbionts, the bacteria manufacturing the compound and sharing it with their host, another benefit of the symbiotic relationship beyond simple light production. These different solutions to the luciferin supply problem demonstrate the multiple pathways through which organisms can achieve bioluminescence. Evolution, finding various answers to the question of how to obtain the raw materials necessary for light production. Each answer creating different trade-offs and dependencies that influence the organism's ecology and evolution. The speed at which bioluminescent reactions occur varies widely, from the flash of a disturbed dinoflagellate lasting a fraction of a second to the sustained glow of anglerfish lures that can continue for hours. Fast reactions serve functions requiring immediate responses, such as startling predators or creating burglar alarms that need to attract attention quickly before threats pass. Slower, sustained glows serve different purposes, including counter-illumination that must be maintained continuously, or lures that need to remain visible long enough to attract prey from distance. The kinetics of bioluminescent reactions depend on factors, including the concentration of reactants, the efficiency of enzymes, the temperature of the environment, and whether the system is designed for burst activity or sustained output. Organisms control reaction speed through various mechanisms, some regulating the availability of oxygen or other required cofactors, others controlling the mixing of separated reactants, still others using neural signals to trigger rapid cascades that propagate through photophores. Understanding the biochemistry that controls reaction speed helps explain why different bioluminescent systems evolved for different functions. The chemistry constraining what's possible, while still allowing considerable flexibility in how light production is deployed. The ocean's bioluminescence thus includes flashes, pulses, steady glows, and complex patterns. Temporal diversity matching the diversity of functions that light production serves. Some shallow water bioluminescent organisms face unique challenges, their lights potentially attracting unwanted attention during daylight hours, when bioluminescence would be visible against sunlit backgrounds. These species often possess mechanisms to suppress light production during the day, venting excess bacteria from light organs, or using shutters to completely hide their photophores when light would create more risk than benefit. The daily rhythm of light management requires these organisms to sense environmental light levels accurately, responding to changing conditions at dawn and dusk, ensuring their lights remain hidden during danger periods, while becoming available when darkness makes them useful. Some species achieve this through circadian rhythms, internal biological clocks that anticipate daily light-dark cycles, while others respond directly to detected light levels, using photoreceptors to sense when conditions become dark enough for safe bioluminescence. This daily management of bioluminescent capabilities adds complexity to the lives of shallow water species, requiring behavioral and physiological adaptations beyond the basic machinery for light production, creating systems that must be not just capable of producing light, but capable of producing it at the right times and suppressing it at the wrong times. The challenges faced by these shallow water species highlight how context-dependent the advantages of bioluminescence become. The same capability that provides crucial benefits in darkness becoming a liability in light. The fossil record provides limited information about the evolution of bioluminescence because soft tissues, including light organs, rarely fossilize, leaving scientists to infer evolutionary history from studies of living species and from the biochemistry of light production. Molecular evidence suggests that bioluminescence has evolved independently dozens of times across different groups of organisms, the genes responsible for light production showing little similarity between distantly related groups, indicating separate evolutionary origins rather than inheritance from luminous ancestors. The repeated evolution of bioluminescence implies that light production provides such significant advantages that natural selection repeatedly favors its development despite the complexity of the required biochemistry. Each independent evolution of bioluminescence required the development of new luciferin compounds, new luciferase enzymes, and new anatomical structures to house and display light, representing considerable evolutionary innovation not just once but many times across life's history. This repeated invention of essentially the same capability through different biochemical pathways demonstrates both the power of convergent evolution and the importance of bioluminescence in certain environments, particularly the deep ocean, where darkness creates strong selective pressure for any adaptation that produces or detects light. Understanding this evolutionary history helps place bioluminescence in broader context, revealing it not as a minor curiosity but as a major theme in life's adaptation to darkness. The quantum efficiency of bioluminescence, the percentage of reacting molecules that successfully produce light, approaches theoretical maximums in many biological systems, with some organisms achieving quantum efficiencies above 90%. This means that for every 10 molecules of luciferin that react, 9 or more produce photons, rather than wasting their energy as heat or through unproductive chemical pathways. Achieving such high efficiency requires precisely controlled reaction conditions, enzymes that guide reactions along specific pathways, while suppressing alternative routes that don't produce light, and cellular environments that protect reactive molecules from interference by other cellular components. The evolution of such efficient systems took millions of years, countless generations of selection favoring organisms whose bioluminescence converted chemical energy to light more completely, the organisms with superior efficiency gaining advantages in competition for mates, success in hunting, or effectiveness in defense. Humans studying these systems hope to learn the principles that allow such high efficiency, potentially applying these insights to develop artificial lighting that wastes less energy, though the challenges of recreating biological precision in artificial systems remain formidable. The near-perfect efficiency of bioluminescence stands as one of nature's remarkable achievements, chemical reactions performing at levels that seem almost too good to be possible, yet occurring in countless organisms every night throughout the world's oceans. The development of bioluminescence in juvenile organisms often lags behind other developmental milestones, young fish or squid developing light organs only after they've reached certain sizes or life stages. This delayed development might reflect the energy costs of maintaining bioluminescent systems, young organisms prioritizing growth over light production until they reach sizes where bioluminescence provides sufficient benefits to justify its costs. In species with bacterial symbionts, juveniles must acquire and cultivate their bacteria after birth or hatching, the process of establishing symbiosis, taking time and requiring the young organism to encounter appropriate bacterial species in the environment. Some species are born with functional photophores, their bioluminescence appearing immediately or within days of birth, suggesting that light production provides critical advantages from the very start of life, perhaps helping young organisms avoid predation during their most vulnerable stages. The timing of bioluminescent development, like so many aspects of biology, represents trade-offs between competing demands, organisms allocating limited resources among many important functions, the balance point between different priorities, determining when capabilities emerge during development. Watching bioluminescence develop in growing organisms reveals how complex capabilities build gradually, requiring not just genetic information, but proper timing, environmental cues, and sometimes partnership with other species. The development of the capacity to glow being as complex as the light production itself, some bioluminescent displays serve no obvious function, leading researchers to wonder whether these represent neutral traits that persist because they cause no harm, or whether they serve functions too subtle or context-dependent for observers to detect. The persistence of bioluminescence, even when no clear benefit is apparent, suggests either that the capability is maintained because it occasionally proves useful, or that the genetic machinery for light production costs little to maintain when not actively being used, allowing it to persist without being eliminated by selection. Some displays might serve functions that become apparent only during rare events or specific conditions that researchers haven't observed, the true purpose remaining hidden because the right context hasn't been witnessed. Other displays might indeed be neutral, evolutionary holdovers from ancestors where they served important functions, persisting in descendants where changed circumstances have rendered them less critical, but not harmful enough to be actively selected against. Distinguishing between these possibilities requires extensive observation across many contexts and conditions, understanding when and whether bioluminescence influences survival or reproduction in ways that might not be immediately obvious to human observers watching from submarines or reviewing video footage. The mysteries that remain in bioluminescence research remind us how much we still have to learn about even intensively studied phenomena, the ocean keeping secrets about functions and purposes that might become clear only after decades more research, or might remain forever mysterious. The neural control of complex bioluminescent displays requires sophisticated coordination, patterns traveling across photophores in sequences that suggest the existence of neural programs similar to those controlling coordinated muscle movements. Creating a wave of light that travels smoothly across an array of photophores requires each light organ to activate in precise sequence, timing controlled by neural signals that propagate through the organism's nervous system with exact delays between each activation. Some organisms can produce multiple different patterns, suggesting their nervous systems contain multiple programs that can be selected based on context, different patterns serving different communicative or defensive functions. The evolution of this neural control likely paralleled the evolution of the light organs themselves, each requiring the other to provide benefits, selection favoring organisms with better coordinated displays and with anatomical structures that made coordination easier. Understanding how nervous systems control bioluminescence provides insights into general principles of neural coordination, how brains and nerve nets achieve precisely timed outputs that create coherent patterns from individually controlled elements. The glowing displays that create such beauty when observed are the visible manifestations of invisible neural activity. Electrical signals translated into light through the medium of photophores thought becoming visible as patterns of luminescence that serve purposes ranging from mundane communication to desperate defense. The ecological impact of bioluminescence extends beyond individual interactions to shape entire marine communities. The presence or absence of light production influencing predator-prey relationships, habitat use patterns, and energy flow through food webs. In regions where bioluminescence is common, predators must adapt to hunting prey that can create sudden flashes or clouds of light, selecting for hunting strategies that counter these defenses, or for predators that can tolerate the temporary blindness that bright flashes create. Prey species in bioluminescent environments must similarly adapt, developing increased sensitivity to bioluminescent signals that might reveal approaching predators, or learning to recognize the patterns that distinguish dangerous bioluminescence from benign sources. The daily vertical migrations that many bioluminescent species perform, rising toward the surface at night and descending during the day, transport nutrients and energy between ocean layers, connecting deep and shallow ecosystems through their movements. Bioluminescence itself consumes energy, organic compounds being converted to light that ultimately dissipates as heat after being absorbed by water and tissues, representing a pathway through which chemical energy leaves biological systems without being transferred to other organisms through consumption. These community-level and ecosystem-level effects of bioluminescence demonstrate that individual capabilities, when widespread, can influence the structure and function of entire ecological systems, the sum of countless individual light-producing events shaping the ocean at scales far beyond any single organism. The future of bioluminescence research includes efforts to use light-producing systems for practical applications, from medical imaging that uses bioluminescent markers to track diseases within bodies, to environmental monitoring, where bioluminescent organisms serve as pollution sensors. The genes responsible for bioluminescence have been inserted into various organisms that don't naturally glow, creating glowing fish, plants, and bacteria that demonstrate the transferability of light production between species. These applications raise questions about the ethics of modifying organisms to produce light for human purposes, concerns about unintended consequences if genetically modified organisms escape into natural environments, and debates about whether such manipulations represent appropriate uses of biotechnology. Beyond practical applications, basic research continues, revealing new bioluminescent species and new mechanisms of light production, the ocean's vastness, ensuring that discoveries await in depths and habitats not yet thoroughly explored. Each new species discovered adds to our understanding of how many ways evolution can produce light, expanding the catalog of biochemical solutions to the challenge of converting chemical energy to photons. The study of bioluminescence thus remains vibrant and ongoing, mysteries yet unsolved, keeping researchers fascinated, applications not yet imagined potentially waiting to be developed, the ancient phenomenon of living light continuing to illuminate both the ocean and human knowledge. The synchronization of bioluminescent displays among groups of organisms creates spectacular collective phenomena, multiple individuals flashing in coordination to produce effects impossible for lone organisms. Some species of ostracods produce synchronized courtship displays, where many males create their glowing patterns simultaneously, the collective display being more conspicuous and perhaps more attractive to females than individual efforts would be. The mechanisms by which these organisms achieve synchronization vary, some apparently sensing each other's flashes and adjusting their own timing to match, creating emergent coordination from individual responses to neighbors, while others might respond to shared environmental cues that trigger multiple individuals simultaneously. The mathematical principles underlying synchronization in biological systems apply to bioluminescent displays as they do to other synchronized behaviors like chorusing in frogs or flashing in fireflies, patterns emerging from simple rules, followed by individuals creating complex collective behaviors. These synchronized displays transform scattered individual lights into coordinated shows, the whole becoming greater than the sum of parts. Biological coordination creating temporary structures made of light that exist only through the participation of many individuals, dissolving when coordination breaks down or participants disperse. Witnessing such displays provides visceral demonstrations of how life creates order and pattern through interaction. Individuals creating something collectively that transcends their individual capabilities. The preservation of bioluminescent capabilities across geological time, through environmental changes and extinction events, suggests that light production provides such fundamental advantages that organisms maintain it despite pressures that eliminate other traits. The lineages that possess bioluminescence have survived mass extinctions that eliminated countless other species, their light-producing capabilities presumably contributing to survival through periods of environmental stress, when any advantage could determine whether a lineage persisted or perished. The stability of the basic biochemistry of bioluminescence across millions of years indicates that evolution found solutions so effective that major improvements became difficult, the system reaching a state of optimization that resists further significant modification. However, the details of light production continue evolving, patterns of photophore arrangement changing, control mechanisms becoming more sophisticated, applications of light production diversifying into new functions. As organisms explore the possibility space around the fundamental capability of producing light, this combination of conserved core mechanism and evolving elaborations characterizes many biological systems, evolution, maintaining what works while continuing to explore variations that might work even better. The persistence of bioluminescence through Earth's changing history testifies to its importance, a capability so valuable that organisms have maintained it through challenges that transformed the planet, light production remaining relevant through epochs, while other adaptations came and went. The sensory experience of bioluminescence, for the organisms that see it, remains largely mysterious to us, as we cannot know what a fish or squid perceives when viewing bioluminescent displays, whether they experience anything analogous to the beauty and wonder that humans feel when witnessing glowing organisms. The neural processing of bioluminescent signals in fish and invertebrate brains differs from human visual processing, their simpler nervous systems extracting different information from light patterns, attending to features like timing and intensity that carry meaning in their world, but might seem less significant to human observers. Some organisms might perceive bioluminescence purely functionally, recognizing specific patterns as signals of food, danger, or mating opportunity, without any aesthetic dimension to their experience. Others, particularly species with complex brains like octopuses or larger fish, might experience something richer, though we have no way of knowing whether their subjective experience resembles anything like human perception. This uncertainty about animal experience extends beyond bioluminescence to all animal perception, the gap between objective description of sensory processing and subjective experience of sensation remaining unbridgeable by current science. The lights that seem so beautiful to human observers serve practical purposes for the organisms that produce them, their beauty perhaps being incidental to their function, or perhaps beauty and function being inseparable in nature, effective designs generating aesthetic appeal as an inevitable consequence of working well. And now, the lights fade behind your thoughts. We've drifted through 100 quiet truths about bioluminescent creatures, from the chemistry that turns life into light, to the depths where darkness blooms with living stars, from the tiny dinoflagellates that sparkle in waves, to the enormous siphonophores carrying constellations through the deep. You are connected to this world. The same chemical elements that produce light in marine bacteria exist in your own cells. The same oxygen that fuels their glow cycles through your lungs. The same carbon that forms their luciferin builds every molecule in your body. The ocean's living lights remind us that darkness need not be empty, that life finds ways to illuminate even the most lightless places, creating beauty and meaning from simple chemistry. Tonight, as you rest, bioluminescent creatures continue their glowing existence in the ocean's depths, flashing signals we cannot read, hunting with private beams of red light, camouflaging themselves against faint starlight filtering from above. They drift and swim through liquid darkness, each carrying its own small sun, writing messages in light that dissolve as soon as they appear, temporary and eternal. The ocean's own poetry written in photons, and you are safe, warm, resting in a world full of wonder, a world where darkness gives birth to light, where the smallest creatures carry stars within them, where the deep ocean glows with life that most eyes will never witness. May their gentle lights guide your dreams, their chemical fires warm your sleep, their patient glow carry you softly into darkness that holds no fear, only rest. If you're still awake, go ahead and watch the next chapter. Sleep well and dream softly. Good night.